everyone, being that you're watching this video, I'm sure you're seeking how to go about starting how to do a transformation using competent cells. So throughout this video, and me personally knowing that transformations do involve a lot of steps, I would like to just go over a step-by-step process with you guys and what is involved in these processes. So feel free to even take notes throughout this process through the various important things that are going to be discussed, or even the important key components to look out for while you guys do your transformations. So now to begin, a basic overview. As mentioned before, transformations do involve the use of competent cells. So when it comes to competent cells, we want to know, what is competence? So overall, competence has been termed to be the cell's ability to take up foreign DNA from its surrounding environment. And how well a competent cell takes up that foreign DNA is related to its competence. So overall, performing a transformation experiment is an important step to take for protein overexpression, which is why doing a transformation is key, and doing it correctly is key as well. So overall, as a safety precaution notice that I want to go over with you guys, the tuner D3 cell line is going to be used. And overall, due to the fact that these cells are of a very weak strain, no other than necessary precautions will be taken. So normal PPE will be used. And what that entails is just the use of gloves, a lab coat, and also goggles. So furthermore, being that these cells are weak, and the fact that we are using cells, sterile technique will be used throughout the experiment to keep the integrity of these cells in order to prevent any contamination from its surrounding environment or from yourself as well. So thank you for sticking with me so far. In this next portion of the video, I would just like to discuss what sterilizing the environment around you is going to involve and how to go about doing that. Preheating the heat block, we're just going to just include what a heat block is in general, why we need to preheat it, and keeping the competent cells on ice. Now, for that portion, we're going to discuss why that's important. So overall, this will be discussed. And yeah, let's get to it. Alright, so welcome to my lab. This is the area where I do my transformations. In order to sterilize the environment to avoid any possible contamination, what we're going to do is wipe down the area with some ethanol. We're going to take the bottle as seen and squirt some on the bench. From there, we can take a few chem wipes and wipe down the surface. And this is going to be done thoroughly throughout the area in which we're going to be utilizing in order to do our transformations. So from there, that is all there is to it, and you can throw the chem wipes in a regular trash can. Alright, so now we sterilize our environment. I wanted to highlight some things regarding the heat block instrument that we will be using. So, on the right we have our low and high button that we'll be using in order to have the heat block on a low or high temperature setting as it heats up. The little bulb here will then light up, which ensures that the instrument is on and beginning the heating process. For the low temperature and high temperature settings, you can adjust that accordingly to the knobs as seen here in blue. Here we have our wells, which we are going to be placing our samples in. The thermometer is placed here in the low heat setting area, as the temperature we will want to heat our samples at will be at 42 degrees C. Alright, so now to help visualize how this occurs, here I am turning on the heat block on the low setting, the bulb lights up, and from there we can take our DI water bottle and then begin to pour some DI water into the wells that we'll be using in order to ensure a thorough heat transfer of our samples. Alright, so now with our heat block instrument preheated and we sterilized our environment, we can now thaw our foreign DNA and competent cells in ice. This will take a couple minutes. Once this is done, we're going to transfer 50 microliters of our competent cells into an autoclaved microcentrifuge tube. In doing this, we're going to use our flaming technique to ensure that our sample remains sterile. You quickly flame the pipette before and after transferring the sample. After that, we're going to add 1 to 2 microliters of DNA to the sample tube using the same technique. From there, the cells and DNA are then incubated together on ice for 10 to 30 minutes. Alright, so now that the incubation has been done, we're going to heat shock our sample for exactly 45 seconds using the preheated heat block. This ensures that the cells uptake the DNA that has been incorporated. The sample goes back directly on ice for 2 minutes. During this time, we'll be preparing to add 150 microliters of autoclaved lysogeny broth, LB for short, using the sterile technique you see here. So now that our LB has been added, there will be another incubation time for a minimum of 45 minutes if it's your own time to a max hour and 30. I usually do an hour. The sample is going to be placed in just something that you might have that will be able to hold the sample like seen here. It will shake for 180 RPM with the set temperature of 37 degrees C. You're going to want to occasionally take the sample outside of the incubator and flick the cells in order to make sure that the cells are all sealed at the bottom. This is usually a great time to warm up our plates that have been stored in the fridge to room temp. These plates contain a 100x stock of ampicillin due to the fact that the DNA used contains genes that are integrated for ampicillin resistance. This will ensure that the cells that potentially grow on our plates contain the desired integrated DNA. From there, during the wait time, you will catch up on studying if you like, or just grab some food, or maybe on both, which is what I'll be doing when I wait. Alright, now here comes the fun part. After labeling our plates, we can then begin the process of adding our sample to the plate. Our plate is going to contain 100 microliters of our sample. To begin, we will use the same flaming technique that was previously seen, but we're going to do something a little different. We're going to use the cell spreader to spread our sample throughout the plate. First, we're going to take our cell spreader that was in ethanol, making sure that there are no drop residues as is flammable, and then quickly flame the spreader over the flame. From there, we will cool the spreader around the areas of the agar plate that don't contain the sample. After this, we can use it to spread the sample while turning the plate at the same time. From there, we will then flame the spreader again and place it back at the ethanol. Overall, this is the stir technique that we'll be using. Okay, so now that we're finally done, we can flip over our plates agar side down to prepare for overnight growth. Thanks for watching. I hope that this video was informative and helpful for you in understanding this process.